Yeah, not bro. Okay then.
Can
You
you
have
decide
to-
what today
Today, brother was Iman. He's on duty.
on duty?
Yeah,
Oh, you make like that.
yeah, we
Very
make
good
a duty
idea.
roster.
A duty roster.
I already set up your live streaming.
Is it?
Yes.
Okay. And can you set up this?
Oh yes, yes. Wait.
Brother, who's that?
Wazif Iman. He is sitting. Oh, he's not here yet.
He's not here yet.
Yeah. Oh, he's at the toilet just now. I asked his friend.
Thank you very
Okay, very good.
Has it?
Yes, has it also.
Sometimes I confuse.
It's okay. You can still use the pointer, but you can adjust the slide because the button is
Losing.
broken. But you can still use the laser. It's here.
No, I cannot move it.
But I cannot move it.
Oh,
I
so
don't
you
know
have
why.
to sit down.
Yes. But actually, we suggest you to use this only because at the students at home, they can see you point here.
Okay. But sometimes, for me, in order to explain well, sometimes we use the whiteboard.
Oh.
Yeah.
So
How
the
many you?
right now, I think 2. Last night 4. Yes,
Okay,
all
they couldn't
sisters.
come.
Oh, they,
Sisters.
yes,
Where
they
are
was
they
from home.
All of them
Kelantan.
from Kelantan.
Yes.
How about the other place like Johor Bahru, Penang?
Oh,
They also affected some area.
I think, but from what I know, is they are all from Kelantan. I
I
think
see.
they come earlier, so they don't get affected because Johor they
Oh,
got
luckily they
affected.
after they come, and
Yeah,
then
like that.
there is flood. So thank you very much for that.
You're welcome.
How's the mouse?
Huh? I didn't check it yet, doctor.
You can check
I'm
in the
nervous.
system. You have to check and keep it the mouse.
Okay, let's do
Yeah. So I tried my best to finish earlier. So within 2 days I try to settle. So 2 days I can announce the result. So that no, I also forgot to do that.
It's okay, doctor.
Maybe the other day.
Thank you, doctor.
Yeah, you're welcome. Okay. Very good morning. Selamat pagi, everybody. Hello, hello. Can you hear me? Yeah, the voice. Hello, hello, hello. Yeah. Can you up the volume a little bit? Sound volume. Okay, brothers and sisters, before we going to start today's class, one representative from the brother, can you recite the word first? Thank you very much, brother. Okay, sisters, brothers, how are you? Yeah, you all are safe, eh? Okay. So today I'm going to discuss, can you hear me? Clearly, right? Okay, so the protein metabolism. So we have already finished the carbohydrate metabolism, fat, lipid metabolism. Now going to start about the protein metabolism, right? Okay, my dear sisters and brothers. Hopefully at the end of this lecture, you will be able to describe the normal pathways of amino acid metabolism, right? And then the important metabolic disorders, yeah. You will have a great chance to learn about the abnormal metabolism as well, metabolic disorders. So whenever we talk about the protein metabolism, so you will be always reminding yourself about the, you know, the food that rich on the protein, right? Rich of the proteins, such as the meat, milk, right? And then the beans, right? Lentil, and then the poultry, right? Okay, my dear sisters and brother. So because the protein is very important for us for the growth and development, and you have already known that protein function as an enzyme that plays important role in the biochemical reactions, right? Protein plays important role as a hormone that regulate the body functions, you know. The protein as immunoglobulin, right? And then albumin and other plasma protein. So they have their own distinctive function in the body, right? So protein is very important. So today I will cover about this subtopic. These are the amino acid pool, protein breakdown, and then the digestion and absorption. And I will introduce you the some metabolic errors related with the protein metabolism. Okay, my dear sisters and brothers, shall we start today's lectures, right? So amino acid pool, actually it's not the real pool, it's imaginary pool. So free amino acid are available, you know, and it's, mainly useful source of the amino acids are available inside and outside the cells. Maybe some of the amino acids are present in the intravascular fluid. Some of them are present inside the cells. So these amino acids, how do they enter into the tissues and bodies, you know, that you consume the protein are digested, amino acid is released, and then they are absorbed through your intestinal tract, and later on they are transported into the liver by the portohepatic circulations. Thereby, the amino acid are accumulated inside the liver in a absorbed state, right? And that amino acids will go to the three different ways. First of all is the amino acid will be used for the new protein synthesis. Some of the amino acid will be utilized for the formation of new product, specialized product synthesis. Some of them will be completely catabolized. So that's why the amino acids are what you call that will be recycled again and again, so that we call it amino acid pool. So you can imagine that here. New proteins are required, especially for the, you know, for the growth and development, you know, and then also some of the specialized products such as the purine, nucleoprotein, pyrimidine, nitrogenous base, heme is made for the hemoglobin synthesis, and neurotransmitters, hormones, you know, all are derived from the amino acid protein hormones, right? And when they are catabolized, they also release the nitrogenous product like uric acid, right? And then also amino acids are fully catabolized as a form of urea. You can see that. Urea are water soluble, can be excreted in the urine, right? Some of the amino acid can transform to the glucose as well. You know, the glucose as well, that kind of amino acid you have already aware about that, right? Glucogenic amino acids, right? They can transform to the glucose. When glucose is formed, the glucose can enter to the tricarboxylate acid cycles and then the glycolysis, right? And we provide the ATP and carbon dioxide is released, so on and so forth, right? So actually, your amino acid pool is maintained in only 100 grams. So amino acid turnover. What is amino acid protein turnover? Protein turnover means some of the amino acids is utilized for these newer protein synthesis. At the same time, some of them are completely degraded. So protein turnover means simultaneous synthesis of the degradation. It remain constant. That indicate that your body's nitrogen balance is maintained. Yeah? Your body is maintained in the nitrogen balance state. So that we call it nitrogen equilibrium. I feel like you cannot hear me well. Can you hear me? Okay, microphone. So there will be the two conditions. We can find in our body positive nitrogen balance and negative nitrogen balance state. So, for example, growing children, they require the additional protein in order to construct the body, bone, muscles, growth and development. So they will always show the positive nitrogen balance. They require protein more, right? And also patients that is recover from some illness, the nitrogen requirement is high. So you can find the positive nitrogen balance in that state. And pregnant women, because in the pregnant woman, inside the womb, there is a growing baby, right? For the development of the growing baby, they require the higher amount of the protein and they create the positive nitrogen balance in your body. Okay, so that's why when you check that, intake and construction of muscles are more than the output. That means some of the protein are stored, right, for the growth and development. So normally, there's someone who lost weight and then the negative nitrogen balance are related with someone who underwent the surgical operation and someone who has advanced cancer, lost weight, and they have Chiastic features and chronic illness like, tuberculosis and then the acute autoimmune deficient syndrome. So their intake will be the less than the destruction of the protein is high, right? My dear sisters and brother. So amino acid metabolism is mainly found in your liver, muscles, right? And then also the waste product of the amino acid metabolism is excreted in the urine. Okay, my dear sisters and brothers. So your amino acid pool is actually, maintained in the steady states in order to maintain your health in an optimal situation, right? Okay, my dear sisters and brothers. So, referring to that slide, you'll be going to study detail about how porphyrin, creatinine, neurotransmitter, nucleotides are synthesized from your amino acid. And amino acid catabolism release not only the carbon dioxide and urea, but also release the uric acid, ketone bodies, and then the glucose as well, right? Okay, my dear sisters and brothers, shall we go detail of the protein digestion and absorption before going to talk detail of the metabolism as usual, right? So dietary proteins are in terms of principle of digestion and absorption. Your protein digestions occur in the stomach by the hydrochloric acid and then the hydrochloric acid and then the pepsin released from the peptide cells. So the protein, you have the protein long chains are degraded and release the small short peptide. And then also when the chyme enter to the small intestine, duodenum and jejunum, what happens is that your pancreas, especially from the exocrine glands, secretes the enzyme, digestive enzyme, right? Digestive juice, right? And then also small intestine also secrete the specific digestive enzyme and complete the digestion of the shorter peptide and finally release the amino acids. So in terms of the digestive enzyme, we simply term protease. The protease enzyme can be categorized into the two types, endopeptidase or exopeptidase. Early digestion of the longer chain peptide. The enzyme plays important role for the digestion of the longer peptide is called the endopeptidase. So they make the longer polypeptide to the shorter peptide and smaller peptide, and then exopeptidase is released from the intestinal mucosa. They digest the tripeptide and dipeptide. Finally, individual amino acid is released at the end of the digestion process. Hopefully you got it. Everybody got it? Okay then. So we can categorize the 3 phases in digestion of the protein. What happened inside the gastric, stomach is called the gastric phase. Because in the stomach, your gastric juice secrete the hydrochloric acid and pepsinogen. A pepsinogen turn again to form the pepsin and digest the longer protein to the peptone and proteose and shorter peptide. Then later, when the food chymes enter into the duodenum, pancreatic juice secrete the specific peptidase called chymotrypsin, trypsin, carboxypeptidase, elastase, so on and so forth. They will complete the digestion, right? Peptide into the amino acid. Some of the leftover dipeptide and tripeptide digestion will be completed by the exopeptidase that is released from the intestinal mucosa cells, right? Okay, you got it roughly, right? Okay, so the detail is that you can see that your gastric juice actually, the watery juice, 97 to 99% are water. The rest are mixed with the hydrochloric acid, mucin, and inorganic salt and digested enzyme. You can see that. So digestive enzyme is pepsinogen, gastrolipase, and renin, right? These are the enzyme that is secreted from your stomach mucosa cells. So you can see that chymosin, known as the renin, that is a specific enzyme that is responsible for the digestion of the mother's milk, especially in the infants. So that the protein digestion and absorption even completed in the infants, infant time, right? Especially mother's milk protein are digested and absorbed in the stomach wall, okay? But there is no longer produced when you turn to the adult, right? So raw digestion is make a curdle or coagulate the milk in the stomach and absorption is start. Okay, so that is about the renin. So now I'm going to focus about hydrochloric acid. Because of hydrochloric acid secreted from the chip cells, what happened is that hydrochloric acid can kill the microorganism. That is, the unfortunately happen in mix with your diet, right? So can kill and also denature the protein and provide the acidic environment so that your pepsin, it's the best optimal pH for the pepsin is that 3 to 4, right? So that's why your stomach will create the acidic environment for your pepsin digestive enzyme. So actually, the pepsin is also secreted from the chief cells or peptide cells. It is actually endopeptidase enzyme, type of endopeptidase. When your pepsinogen is secreted from the chief cell, it is a very long chain. We call it pepsinogen. We also call that zymogen granules. It is an inactive form of the enzyme. So after removal of the 44 amino acid sequence from the N-terminus, do you remember that? In the peptide chain, there is an amino terminus and carboxy terminus, right? Amino terminus side, there are 44 amino acids are removed from the pepsinogen. Pepsinogen chain becomes shorter, right? The shorter pepsinogen is called pepsin. This is the active form of the enzyme. So pepsin try to hydrolyze the protein and degrade the peptide and some of the amino acid. So this is the digestion completed in your stomach. Okay, my dear sister, brother, you got it right. Shall we move to the pancreatic phase, right? So shall we go along with the food chyme? Now food chyme is already inside the your duodenum, right? So pancreatic juice is secreted from the pancreas, exocrine part of the pancreas gland. And then so it contains the bicarb. So could you imagine that your pepsinogen mixed in your food chymes is automatically inactive because of the changes of pH, right? So optimal pH for the pepsin is 3 to 4. Now the pH changed to the alkaline pH because of the bicarbonate secreted from your pancreas. Pancreas. So pepsin will stop its function. So other proteases secreted from the pancreatic acidic cells are chymotrypsinogen, proelastase, procarboxypeptidase, and then the A and B. So first of all, before going to talk about this enterokinase enzyme, I have to mention about the pepsinogen. That pepsinogen, I told you, right? 44 amino acid is trimmed, right? Amino acid are trimmed and degraded from the amino terminus. What are the factors that cause the trimming process? At the beginning, hydrochloric acid itself degrades the 44 amino acid from the pepsinogen. Make the shorter pepsin. Okay, once hydrochloric acid done, the rest part is that pepsin itself degrade the pepsinogen, 44 amino acid, amino sequence of the pepsin. So pepsinogen itself make the pepsin. Pepsin itself make the pepsin from the pepsinogen, removal of the 44 amino acid. So that kind of mechanism is called autocatalysis. Autocatalysis, self-killing, suiciding. Pepsin suicide the pepsinogen, right? Yeah, to make it more pepsin. So that reaction is autocatalysis. Got it, right? Okay, autocatalysis. Catalysis means reactions, right? Reaction. Auto means pepsin itself degrade the 44 amino acid sequence of the pepsinogen, right? So to make the pepsin more pepsin, right? Everybody got it? Yes. Okay. So enterokinase and enteropeptidase is secreted from the duodenal epithelium. What is its function? It removes the amino acid change from the proenzymes, make them become an active form of the enzyme. Okay, proenzyme. Okay, first of all, enteropeptidase degrade the trypsinogen by removing 6 amino acid residue from N-terminal part of trypsinogen. I repeat again. 6 amino acid residue from the N-terminal part of the trypsinogens are removed. Trypsinogen will change to the trypsin. This shorter and active form of the trypsin again can activate to the other proenzymes, chymotrypsinogen, proelastase, procarboxypeptidase, to form the active form called chymotrypsin, elastase, carboxypeptidase A and B. Okay, got it? So each enzyme has their own property and their own function. So they will attack to the peptide bond, specify for the side chain group of the particular amino acid, okay, that adjacent to the susceptible peptide bonds. For example, let's see, you can see that the trypsin attack to that peptide bond. Can you see that? So arginine and lysine are removed because the trypsin recognize their side change. So break the peptide bond adjacent to these amino acids. So these amino acids are released from the peptide. Chymotrypsin attack to the tryptophan, tyrosine, phenylalanine. Do you remember that? That is an amino acid that contain the ring structures in their side chain. It's attacked by chymotrypsin. Elastic attack the peptide bond adjacent to the alanine glycine serine. So carboxypeptidase A and B attack to the peptide bond adjacent to alanine, isoleucine, leucine, valine, so on and so forth. And B attack to the arginine and lysine as respectively. So that means that chymotrypsin doesn't digest the peptide bond adjacent to the leucine or valine, right? They have their own specific digestive enzyme work, right, to digest the peptide bond. Everybody got it? So finally, as a result, you will receive the small peptide and then the amino acid. The small peptide will be degraded by the dipeptidase and tripeptidase. They are called the exopeptidase. And finally, all the protein are degraded. The protein and the proteases digested, and all the protein are degraded and release the amino acid. Right? Right. But there will be the stay left over. Few dipeptide and tripeptides are still available. Don't worry, they can be absorbed. Right? This is about the digestion of your protein. Yeah. Whenever you eat the protein like meat, the fish, egg, all will be digested in that way. Right? Okay, how about your egg protein? So your egg is, there is a two-part, right? Make egg white and egg yellow, right? Egg yolks. So egg white is completely protein in nature. So they will undergo the protein digestion, the procedures. But your egg yolk, yellow color, is mixed with the nucleic acid, right? So they will be digested in different way. Okay, so the egg yolk consists of DNA and RNA. You can see there, DNA and RNA. So your gastric pH denature the DNA and RNA present inside the egg yolk. And then the denature the nucleic acids, the food when enter into the intestine, duodenum. The pancreatic juice releases the nuclease enzyme. Okay, nuclease enzyme degrade the long chain polynucleotide to the oligonucleotide, smaller nucleotide, and phosphodiesterase enzyme will remove the phosphate. You can see that mononucleotides. Mononucleotides remove the phosphate again. So this phosphodiesterase enzyme break the phosphodiester bond. You cannot see there. You can see here, my dear sisters and brother. This is nucleotide. Your base attached to the sugar, and sugar attached to the phosphate, and make the change longer. This is a structure of your DNA, right? The base attached to the sugar and phosphate. Phosphodiester bond make the change longer and make the polynucleotide. Right now, first of all, denature this hydrogen bond so that the double helix structure is, detach each other. And then phosphodiesterase enzyme break the phosphodiester bond. And finally, you will receive the mononucleotide. Mononucleotide consists of base, sugar, and then the phosphate, right? Phosphate will be absorbed in phosphate transporter system. Sugar, ribose sugar, is transported through their own transporter system. Do you remember that? Glucose transporter system, right? And the base has their own transporter system. Finally, they all are absorbed, right? when they reach to the mucosa cell, the nutrients are transported to the liver via the portohepatic circulation. So far, everybody got it? So you can categorize the digestion into the two types, depend on the food, right? The protein-rich food A and nucleoprotein-rich food is a totally different, right? Digestion, okay? Okay, my dear sisters and brother. So your protein absorption is almost similar like your carbohydrate digestion, glucose absorption. Your absorption of the protein, absorption of the amino acid the method is more or less the same like glucose absorption method. It's different from the lipid particle absorption, right? So lipid particles, you can see that it's an emulsification of the droplet. Later on, they undergo the micelle formation. The micelles are endocytosed and enter into the mucosal cell, right? But your protein absorption is like your glucose absorption. You must have their own transporter system. You must have own transporter system. So there are various types of amino acids, right? In the protein digestion, as an end product, they are transported via their own transporter system. There are 6 major transport systems for the different amino acids. Okay, so absorption takes place in the duodenum and jejunum. Neutral amino acids are taken up by their own transporter. So example are serine, threonine, and alanine. Whereas ineffective aromatic amino acids have their own transporter system. They are absorbed through their own transporter system and enter to the mucosal cell. Amino acids such as proline and hydroxyproline, they have their own transporter system, used to absorb into the mucosal cell. So beta-alanine, beta-amino acid, taurine are transported via their own transporter system. Similar ways goes to the basic amino acids. Cysteine, arginine, ornithine, histidine, and lysine, they use their own transporter to enter into the mucosal cell from the lumen. Acidic amino acid, you know that, right? Aspartate and glutamate, they use their own transporter system. And finally, they enter into the mucosal cell. So when you check that, what are the type of the transport? Okay, facilitated diffusion. At the beginning of the absorption, there is in terms of concentration gradient, amino acids concentration is higher in the lumen than the bed of concentration inside the mucosal cell, right? So the facilitated diffusion method is used, and then they enter into the mucosal cell. So once concentration gradients are the same outside the cell, in the lumen and inside the cell, they will use the sodium potassium acid transporter system. This is a co-transport. You can refer to the glucose transport, right? So a molecule of sodium enter in a line with together with the molecule of amino acid will enter into the cell via the acid transport process, sodium-dependent acid transport. And dipeptide and tripeptide, they have their own transporter, so they are transported via their own transporter. So when they reach inside the mucosal cells, all the peptides, small peptides, that is recently absorbed will be completely digested into the amino acid. That amino acid will be reabsorbed in the portohepatic circulation. So all the amino acid will be transported to the liver, right? Liver, right? Okay. Any questions so far? Any questions from the brothers, sisters? Any questions from online live stream student? Can you hear me? If there is a questions, let me know. Okay. Now, okay. So your amino acids are transported into the liver now, right? Okay. So in order to enter into the liver cells, there is also transporter system, yeah, that we call is gamma glutamine cycle. Gamma glutamine cycle, you know that? You know that glutathione carry the amino acid and gamma glutamine amino acid, you know, the deload the amino acid inside the liver cells and they recycle to carry the amino acid that come from the blood circulation, right? That is carried in the circulation, right? You can see that. This is also ATP dependent transport. Everybody got it? Okay, so far, right? Now, special for the pharmacy professionals, I need to introduce you with the one condition that is we call celiac disease. Celiac disease is a genetic autoimmune disorder in which your intestinal villi are damaged. Yeah, Dr. Azra said that you haven't started the GI tract yet. GI tract, gastrointestinal system will start in next week, right? So, but listen carefully, my dear sisters and brother. Actually, this disease patients, what happened is that they are hypersensitivity reaction. The protein is called gliadin protein. The gliadin protein is derived from the gluten. The glutens are rich inside the wheat, barley, and rye, and also rice, right? That gluten, especially the gliadin protein, is hypersensitivity, you know. So hypersensitivity, they alter the immune system. Immune system responds to hypersensitivity. So finally, the immune system attack to the finger-like villi, small intestine. Normally, your small intestinal villi look like that, very tall, and it's like, they can move, and they help the peristaltic wave. You have a peristaltic wave. Now, they all flatten. So the damaged villi doesn't work, So as a result, what happened is that someone who are having this disease, hypersensitivity of the gliadin, gluten sensitivity, they will present with the malabsorption syndrome. Absorption's impaired. As a result, they will got the loose motion, diarrhea, the wind, the bloated, and then some of them will present with the constipation. What happened is that the persistent and unexplained GI symptoms such as they usually feel like nauseated and vomiting, stomach pain, and also cramp, and then the bloating feeling. Of course, the milk absorption occur as a result. Vitamin absorption also impaired. Mineral absorption also impaired. As a result, they will complain of with the iron deficiency, vitamin B12 and folic acid deficiency symptoms. They used to have a tiredness, headache, and weight loss, right? It's related to the your gluten, the your gluten consists of gliadin protein. So that protein causes the immune system irritation. As a result, respond back to the immune system, right? Your intestinal villi got damaged, right? So, and another disease condition that I would like to introduce you is the cystinuria. Cystinuria refer to your amino acid cysteine. My dear sisters and brother, your nephron, have you ever got the urinary tract system in physiology? Already got, right? Not yet. Okay, never mind. This is the structure of nephron in the kidney. What is the function of nephron is there? It's filter the blood. So nutrients are filtered. So amino acids are filtered and enter to the proximal convoluted tubule, and loop or Henle, distal convoluted tubule, so on and so forth, right? In the kidney, what happened is that, can you focus to this area? There is a transporter system. Their function is that they reabsorb the filter amino acid back to the systemic circulation. Everybody got it? Everybody got it? Okay then, if you got it, I will repeat. I won't repeat. Okay, you can see that cysteine, ornithine, arginine, lysine. These amino acids all use this transporter system. So the transporter system reabsorb back the filter amino acids that are present inside the lumen to back to the systemic circulation. Now the problem is that the transporter system is damaged or maybe genetically malfunction. The synthesis is not complete. So finally, these amino acids cannot be reabsorbed back to the systemic circulation. As a result, they will be accumulated outside in the lumen. They will create the problem. The problem is that, inability to reabsorb this cysteine will lead to the accumulation and subsequent precipitation leads to the stone formation, cysteine stone formation. Okay, at the beginning, cysteine, higher amount of cysteine is released in the urine. That condition we call it cysteinuria. But cysteine accumulated and precipitated and finally formed the stone formation. So you can see that the cysteine stone here, cysteine stone here. There are various types of stone, a struvite stone, oxalic stone, uric stone, so on and so forth, right? So you can see that this kind of cysteine stone formation mostly affect to the male, okay, male gender. Could you imagine that someone who got a renal stone, after removal, renal stone keep coming, new stone is a keep forming, removal, second time, third time, fourth time. So whenever you can visit to your clinical posting, you may see such kind of case as well, right? So this is related with the genetic problems. Yeah, that genetic problem is affect to the transporter system. The impaired transporter system unable to reabsorb the filter amino acid and create the stone formation. Right? So when you check the inherited disorders of the amino acid metabolism, cysteinuria, cysteine stone formation is very common one. So mostly affected to the male, especially reproductive age male, age around 20 to 40 is a very vulnerable group. They used to get this stone, right? And second problem is inherited disorders found, after the cysteinuria is cysteine. The problem is that there is an enzyme that catabolize the histidine amino acid are genetically deficient. So that's why histidine accumulated and it will appear in the blood. So that condition we call is histidinemia. Histidinemia is a term that is defined for the someone's whose histidine level, plasma histidine level is higher than the normal reference value, right? And phenylketonuria is a quite common disease. So phenylketonuria screening test is mandatory, especially for the developed country like US. They detect the phenylketonuria even in the embryo life. They will, take some amniotic fluid from the pregnant lady and check the cytology and can detect the phenylketonuria, because you know why? The baby who's are born with this genetic disease cannot study because their intelligence quotient is very low, right? And also may have some central nervous system neurological symptoms, so they are unable to develop like other normal children. And After phenylketonuria, methylmalonyl-CoA mutase enzyme deficiency is genetic disorder is common, and albinism. Hey, see? Albinism. Now I got a normal one, right? Normal sound. Okay. So albinism is the situation where the person lost their skin color. Skin color. There is no melanin formation. We call it albino, right? Have you ever seen the albino? Yeah. But albino lacking of the melanins. There are uromelanin, pheomelanins. There are various types of melanin. They lost the melanins. The pity albino, if they are born in certain countries in Africa, there is a myth that the albino body, the skins, they burn the skin and they use the traditional medicine, something like that. So their life is very dangerous, yeah, and danger. Albino, right? You can read that so many stories there. Okay, and then uncommon other type of genetic disorders related to the amino acid metabolism are homocystinuria, acetonuria, maple syrup urine disease. Wow, sounds strange. What is it? So actually, the urine smells like maple syrup. You know why? The certain amino acids are leak out, and also your urine, your urines are full of with this amino acid, especially aromatic amino acids. They create the smell, sweet smell, right? Ketogenic amino acid like phenylalanine, leucine, lysine, presence of them that create the maple smell, right? Maple syrup smell in urine and cysteine thioninuria. So these are the conditions. Okay, my dear sisters and brother, we still have time, right? Everybody got it? Okay. So now your protein is already, your amino acid is inside the liver. Later on, liver transported the amino acid to muscles, right? And then the whole body, right? Because your amino acid is very important for enzyme synthesis, right? And then also important for the hormone synthesis and all that, right? So actually, your protein turnover is simultaneous synthesis and simultaneous breakdown of the protein, right? That utilize only 1 to 2% of total protein mass, right? Especially from the muscle. Okay, let's say protein turnover, 70 to 80% of amino acid will be reused for the new protein synthesis, whereas the rest, 20 to 25% or 30%, will be completely degraded, and urea will be released and removed from the body in the urine. So degradation of the proteins, like us, that RBC only can live only 100 to 120 days. Similar ways goes to the protein as well. Your protein, some of the proteins are degraded very quickly once they've done their function. Some of the proteins, such as your, the collagen fiber, can live longer, months and years. So depend. The lifespan is totally different. Depend on the structure of the protein, depend on how do they function in the body, right? Okay, my dear sisters and brothers. So that's why protein degradation can be categorized into the two parts. So protein degradation for short half-life protein is different and long half-life protein is different. So they all can be degraded by the two pathways, two different pathways. There is ATP independent pathway that is degraded by the lysosomes. Lysosomal degradations can be categorized into the two, again, subcategory, autophagy process and heterophagy process. Autophagy process and heterophagy. Autophagy means intracellular degradation of the protein by the protein. So could you imagine that protein eats the protein? Protein eats the protein. It's because suicidal, right? Yeah, protein eats protein inside the cell. That process we call is autophagy process. Autophagy process. I cannot recall the name of the Japanese Nobel Prize winner who discovered about autophagy process. Those who are fasting, the protein, autophagy proteins are accelerated, it kills the proteins. Especially they digest the H protein and function protein, they degrade it and so that we will get some of the regeneration process decrease. Regeneration process decrease that give us live longer. So you can imagine that they used to do the fasting, intermittent fasting. Those who are really adhere to intermittent fasting can live longer and stay away from the certain chronic diseases. So it is related with autophagy process. Okay, autophagy process means that they damage and some proteins that then their functions are degraded by specific enzymes outside the cell. That is because heterophagy process. So that's why protein lifespans are variable. Some of them are short, some of them are long, some of them are intermediate. Right? Okay, everybody, you got it. And then the short half-life protein is utilized as second pathway, that is ATP-dependent ubiquitin proteasome system. Come again? Ubiquitin proteasome system. That system utilizes the ATP, energy-dependent pathway, and then degrade the short half-life protein. Okay. So, types of the protein catabolism is roughly, you can see there, this is the, the receptor-mediated endocytosis process. Extracellular protein are engulfed and taken up inside the cell. Later on, it is degraded by the lysozyme. You can see that. And some of them enter into the cell by the pinocytosis and phagocytosis. So, what is a phagocytosis? Phagocytosis is eating. Cell eat. Pinocytosis means cell drink, right? Drink together with the fluid, right? The protein together with the fluid inside, lysozyme degraded, right? So autophagy process. So now I'm going to focus about the short half-life protein. How you can categorize, the protein, whether they are short half-life or long half-life, right? So it depends on the structure. So some of the short half-life protein, they last only 3 minutes inside our body. Then they are degraded by the proteolytic enzymes. Some of them can live more than 60 days, some of them year, especially can refer to the collagen fibers. So half-life is depend on the presence of amino acid residue that found in the N-terminus part of the residue. For example, serine is present in the first amino acid of the N-terminus part of the particular protein that consider it's a little bit longer. The lifespan is more than 20 hours, but your N-terminus part of the particular protein, amino acid residue is aspartate. That protein is considered very short half-life protein, especially enzyme, right? You know that, right? Glycogen synthase enzyme, right? Synthase enzyme become active in the welfare state. During the fasting time, glycogen synthase become inactive, right? And some of them is totally degraded, right? So in that case, that enzyme might have this amino acid sequence inside the structure. Okay, that protein contain PAST sequence. What do you mean by PAST? Proline, glutamate, serine, and threonine are present inside the short half-life protein. That test sequence can be recognized by your ubiquitin protein, specialized protein. So once they recognize, they will bind to the targeted protein and completely digest and degrade. Next step. My dear sisters and brothers, you can see there, this is a enzyme. We call it ubiquitin activation enzyme. See, protein, protein bind to the ubiquitin by the process, ubiquitin activation process. Now you can see that ubiquitin is a green color. This process require ATP. ATP dependent binding make the E1, right? E1 ubiquitin complex. The transacylation process occur, meaning addition of the acetyl group attached to E1. E1 will change to the E2 structure. Structural changes will make another second acetylation process, make the E3. So once E3 is formed, the E1 is actually activating, okay, adding the ATP to the ubiquitin. Function of E2 is accepting the acetyl group and making the conjugation of the enzyme. E3 is ligase. Ligase function means is tightly bind to the ubiquitin. Tightly bind to the ubiquitin. So this process is called the ubiquitination. In other words, it is also known as ubiquitinylation process, the same. So now your ubiquitins become activated. Once it's activated, they will bind to the targeted protein. Targeted protein means your short regulatory protein, right? Short regulatory enzyme, H enzyme, once they done the function, the body, we do not require anymore, they will be completely digested, right? So there's the ubiquitin here, green color will bind to your substrate protein. Finally, the substrate protein is what you call that, degraded by your proteasome. As a result, proteasome degrade your substrate protein to the shorter peptide, and then the ubiquitin will be detached, and that ubiquitin will be recycled for the next reaction. So that's why, my dear sisters and brother, you can summarize it. First step is ubiquitin must be activated by ATP, by the enzyme ubiquitin activating enzymes. Later on, activated ubiquitin will bind to your targeted protein, especially, you can see that ubiquitin is orange color, your targeted protein is the blue color, especially the glycine amino acid residue of the ubiquitin. Ubiquitin itself is protein, do not confuse that. So the glycine will attach to the lysine amino acid residue of the target protein. They tightly bind it. The ubiquitin targeted protein complex is engulfed by the proteasomes. Proteasome degraded your targeted protein. Targeted protein become the short peptide. Short peptide is completely digested by the non-proteases enzyme. So as a result, amino acid is released. Okay, you got it, ubiquitination, right? Ubiquitination, this is very commonly asked questions, right? Ubiquitination, ubiquitinylation. Okay, my dear sisters and brother, everybody got it? So I still have 2 more minutes. Let me summarize it, today's lecture. So your amino acid pool. Amino acid, not really like pool, like real pool, but amino acids are accumulated inside the cells, tissues, right? And then also accumulated in your blood circulation as well. These amino acids are used for the, these amino acids are used here for the other amino acid biosynthesis. For example, phenylalanine is an amino acid that is used for the tyrosine, non-essential amino acid. You remember that? Okay, you don't remember, no problem. The amino acids are completely metabolized and release the alpha keto acid, right? Alpha keto acid is corresponding alpha keto acid. For example, alanine can be transformed to the pyruvate. Oxaloacetate can transform to the aspartate. Aspartate can transform to the oxaloacetate because alanine is amino acid, pyruvate is alanine corresponding keto acid of the alanine, so on and so forth. So alanine, when it's removed the amino group, it will change to the pyruvate, so on and so forth, right? Okay, so the degradation of the protein is done by there are two processes, ATP dependent degradation process and non-ATP dependent. ATP dependent is ubiquitin process. Ubiquitin process requires the best sequence of the targeted protein. Best sequence are recognized by the ubiquitin. Then proteasomes degraded and release the amino acid. That amino acid will be recycled for the simultaneous synthesis and degradation, and some of them are completely digested and released to urea and carbon dioxide, right? Some of them are used for the specialized products such as purine, pyrimidine, and neurotransmitter, melatonin, thyroid hormone, and melanin, skin coloring agents, and all that. They all are derived from the amino acids, right? So tomorrow I will explain detail about how the ammonia released from the amino acid in the body. So amino group of the amino acids are removed. Ammonia is released, right? Generated. That ammonia will enter to the liver, urea cycle, and transform the urea, right? So before going from the urea, the amino acids usually undergo the transamination reaction. So that transamination reaction is mainly catalyzed by the two different types of transaminases, which are called the alanine aminotransferases and aspartic aminotransferases. Enzymes. Okay, so tomorrow I will explain in detail about how ammonia is released and how ammonia transforms to the urea, right? So what is the urea cycle? What are the factors that affect on the plasma urea cycle as well? Right? But today I provided some sample questions for you to study and prepare for the exam. Is that briefly explain the digestion of the dietary protein, explain the absorption of amino acid, okay, from the GI tract, and ubiquitination process. So for the MCQ, you need to know about the severe disease, what is the problem related with the severe disease, and what is the problem related with the cystic stone formation, cystic area, right? Okay, that's all from me. Thank you very much. Thank you very much. Any questions from the online student? Any questions? If you have a questions, feel free to ask me, okay? So thank you very much, everybody.
More than output. What does intake means? We're talking about muscle or protein. What?
Intake means the food you eat.
Or
Protein intake. Protein intake.
Okay.
So, yeah. Let me explain to other people. So, no, you can see here. Regarding the positive and negative nitrogen balance, there are two spots is there. Okay. Intake and output refer to the how much protein you eat, meaning the amount of protein Is it 100 gram or 600 gram or whatever that protein intake? Outwardly, the protein will be utilized for the new protein, muscle protein synthesis. Some of them are completely degraded and removed from the body, right? In certain diseases, especially chronic kidney disease, no matter how you see the protein, all the protein are removed in the filter urine. That condition we call is proteinuria. Protein is lost from the kidney because your kidney is failed to function, reabsorb all the amino acid back to the systemic circulation, right? So in case of the growing children, pregnancy, right, the intake must be higher than the output. So that's why their protein requirement is high. They must have a positive balance, right? They must have positive balance. So that the embryo will be growing well, to achieve the target body weight, right? And then also the convalescent patients, for example, they need the more protein for the wound healing process, right? So they must have positive nitrogen balance. Sorry? Dietary protein. Dietary protein is related to the input to the protein input, amino acid input to the muscles as well. Right? Sorry? Yeah. So it's like energy intake and energy output, right? Okay. Negative balance means output is high, intake is less, so that's why they always show that they cannot maintain the equilibrium state of nitrogen balance. Okay? Everybody got it? Okay. Thank you. Dr. Azra is here.